Good afternoon. I'm Jeff Allen, President and CEO of Friends of Cancer Research. Thanks so much for joining today's meeting. We're very excited to have this opportunity to share with all of you an update on a project that we have been working on for quite some time with a wide variety of various stakeholders. We hope that today will provide an opportunity to share some of those updates, but also hear from all of you. We're very excited to have this chance to have what we hope will be an even more interactive opportunity than some of the prior sessions that we've had on this topic of utilizing ctDNA as a biomarker in developing new cancer treatments. We have an exciting agenda for today, which will start with a presentation from some of our lead scientists, including Hilary Stiers and Brittany McKelvey, who will walk through some aspects of projects that we've been collaborating with a large number of experts on related to the potential utilization of ctDNA, followed by an expert panel, which we thank them for joining us today to share their additional expertise about the opportunities of where this field may move in the future. And we certainly want to hear from you and very much look forward to the discussion today. So without further ado, I think we'll go ahead and get started. Thanks for joining us.
There is an exciting new breakthrough in how we treat cancer. Imagine we could obtain vital information about a patient's tumor from a blood sample. Scientists discovered that as tumors change and their cells die, small pieces of their DNA are released into the bloodstream. These fragments are known as circulating tumor DNA, or ctDNA. So what does this mean? Clinicians will be able to identify mutations or alterations in cancer-related genes using a simple blood test. This allows patients to receive more targeted treatments while also monitoring their response to ongoing treatment. That means less invasive testing, more precise and actionable results, and better patient outcomes. This is only the beginning. For this breakthrough to touch as many lives as possible, we will need a large-scale coordinated effort to prove it works. That is where Friends of Cancer Research is leading the charge, bringing together an unprecedented group of stakeholders to propel this tool to the forefront of cancer medicine. Together, we're understanding how ctDNA aligns with treatment outcomes and how it could revolutionize our approach to patient care and accelerate drug development. And we're just getting started.
Hi, everybody. I'm Hilary Stiers. I'm a science policy analyst here at Friends, and I'm joined by my colleague, Brittany McKelvey. Thank you so much for joining our patient advocate webinar. Today, we're going to be discussing some background on ctDNA as a biomarker in cancer development. Follow along with the conversation on Twitter using the hashtag ctDNAFriends. ctDNA is a tool that helps us to understand tumors. So we wanted to provide everyone with some background about how ctDNA is used as a tool. And in the clinic, blood is drawn from the patient, and the sample is called a liquid biopsy. A liquid biopsy is just a test on a sample of blood to look for cells from a tumor or for pieces of DNA from tumor cells that are circulating in the blood. This blood is then passed on to a scientist, and that scientist removes DNA from the blood to test for ctDNA. The ctDNA is identified from that pool of DNA using computer algorithms. The reason that this process works is because DNA alterations unique to a tumor are also found in ctDNA. Theoretically, the amount of ctDNA correlates with the size of the tumor. And the amount of ctDNA depends on a variety of factors, including the tumor type, patient age, treatments received, and more.
So how can this exciting new technology be applied in oncology drug development? Let's walk through the drug development process. So drug sponsors or pharmaceutical companies who have an exciting new novel therapy to test perform clinical trials, which are protocol-driven research studies to generate evidence on the safety and efficacy of their drug for FDA approval, as FDA reviews all oncology drugs to ensure that they're safe and effective for patients. Efficacy, a clinical trial must show that a drug has a clinical benefit, And in oncology, this tends to be showing that the drug increases overall survival in patients. FDA can sometimes conditionally approve a drug if it treats a serious or life-threatening disease. And this pathway for conditional approval is called accelerated approval. Accelerated approval, a sponsor has to show a benefit via a surrogate endpoint, which is an outcome that's measured in a clinical trial, and it has to be shown through data and evidence to be reasonably likely to predict clinical benefit. So that surrogate endpoint itself may not directly show clinical benefit. However, after accelerated approval, a sponsor still must perform additional studies to demonstrate the actual clinical benefit of a patient on the drug to receive full approval. So to look more into these surrogate endpoints, a surrogate endpoint is an early endpoint that predicts long-term clinical outcomes like overall survival. And these are important in the early readout of a drug for potential efficacy. As you can see depicted here on this clinical trial timeline, from the start of a patient receiving an investigational therapy, a patient has to be monitored for a significant amount of time for the trial to reach its primary endpoint and assess overall survival for the drug's benefit. To help give life-saving treatments to patients, we can use early endpoints, which are analyzed earlier in a clinical trial to help correlate with long-term. So here we're looking at progression-free survival, which I'll speak to more on the next slide, which is conducted through looking at a patient's scans and imaging to look at their tumor burden. However, which we'll discuss today, there may be the potential to use even earlier endpoints that can be done, such as ctDNA, that correlate then and predict long-term clinical outcomes for patients. So as I noted, research has to be done to be able to develop the data and evidence that's necessary to support the use of these surrogate endpoints that are reasonably likely to predict long-term outcomes. And I'll walk through here just a few examples that you may be familiar with that are used in oncology as early endpoints. These include progression-free survival, which is the time from when a treatment starts for a patient to then the disease progresses. And this endpoint is used in many different solid tumors. There's pathological complete response, or PCR, which is when you look at the tumor tissue that has been removed after a treatment to see if there's any signs of remaining cancer in that tissue. And this is used in breast cancer. And lastly, used in blood cancer, we have minimal residual disease, or MRD, which is looking in the blood to see if there's the presence of a small number of cells that remain after a treatment. And so with all of these being actively used in oncology drug development, the question remains if we can then use ctDNA as an early endpoint as well. FDA creates documents, and they're called guidance documents, to help explain their current thinking on a topic for drug developers and diagnostic developers. And FDA actually recently released draft guidance about the use of ctDNA in May and how ctDNA may be used in oncology drug development, specifically for solid tumors. And in this guidance, they highlighted 4 potential uses for ctDNA, and I'll briefly walk through these use cases. The first use case is for patient selection. And so that's looking at the patient's blood and their ctDNA to understand if they have a specific genetic alteration that there may be a targeted therapy for that specific alteration that could be used for their treatment. And so if the patient then does not have that specific alteration that correlates with that targeted therapy, then they could be chosen to not receive that therapy. You can also then use ctDNA for patient enrichment. And so this is based off of studies that have shown that the presence of ctDNA after a patient has surgery to remove the bulk of their tumor or after certain therapies in the adjuvant or neoadjuvant setting, that if there's remaining ctDNA, this suggests that the patient may have a higher risk cancer or a higher risk for recurrence. And so if we can identify these patients by ctDNA that have this higher risk, we can then enrich for these patients in a clinical trial to try for new novel therapies. Also, for using ctDNA, we can use it to potentially measure response to treatment. And so this is research that's underway to understand if we see changes in ctDNA or ctDNA dynamics that are reflecting the dynamics or change in a tumor. So for example, before a treatment, a patient may have a large tumor, which is shedding a lot of ctDNA, and high ctDNA levels suggest the tumor's presence. However, after treatment with a therapy, potentially if those ctDNA levels decrease, that would suggest that the tumor is no longer present or has shrunk in size. And so we can use this readout to potentially understand how the tumor is responding to treatment. Along with this then, and measuring response, is the potential to use ctDNA as an early endpoint. So again, being able to look to see if that patient has decreases in ctDNA in response to treatment may suggest that their patient will have a long-term clinical response. And so using changes in ctDNA to then predict that long-term outcome of overall survival for the patient. For different use cases. There's been a lot of data and evidence that are beginning to be generated, especially on the left side here for patient selection and enrichment, which are actively being used in oncology drug development and clinical trials. However, there's still data and evidence that are needed to support using ctDNA to measure response to treatment and for use as an early endpoint. And so our goal here today is to discuss how we can reach this goal, to be able to use ctDNA as an early endpoint to predict long-term clinical outcomes, such as overall survival and progression-free survival, to help get life-saving treatment to patients earlier. And so to be able to reach this goal, again, we need to be able to have the data. And so one way to have the data is to potentially have each individual company, as they're running their clinical trial, collect data on ctDNA levels and patient outcomes. But this has the potential to be very slow. And so our approach that we've taken here at Friends, and that Hillary will speak to in a minute, is to be able to bring together multiple data sources across different clinical trials and disparate data sources together in what's called a meta-analysis. And a meta-analysis is an approach to systematically be able to assess the data from these multiple clinical trials all at one time to help ensure that the effect we see, if we see ctDNA predicting long-term outcomes, exists across all trials in a much faster and aligned way across sources.
And so our meta-analysis at Friends is called CT Monitor, or ctDNA for monitoring treatment response. And the objectives for this project include investigating the feasibility of harmonizing ctDNA, aligning on a methodology for the meta-analysis, and characterizing associations between ctDNA values and tumor response. This project is broken down into 2 steps, and step 1 was completed and a manuscript of findings should come out any day now. Step 2 is ongoing, but first I wanted to describe our approach to step 1. This unique partnership allows for collaboration that might not otherwise have happened. And so our multi-stakeholder group includes statisticians, clinicians, and researchers from academia, industry, and government. I want to say thank you to all of our collaborators. The working group implemented this project together. And the first thing that we did was host a public meeting to identify the key questions that we wanted to ask during this project. The first question we identified is, do ctDNA levels reflect the therapeutic effect of cancer therapies? And the second, can we bring data together? So our working group members then focused on key decisions, including identifying the data sets and sponsors that would be included in the meta-analysis and selecting an independent analysis center. In this case is CRAB, or the Cancer Research and Biostatistics Group. During our analysis, we focused on non-small cell lung cancer treated with immunotherapy, and I'll give you more details about what the process looked like in the next slide. Finally, we reported our findings at a public meeting and in peer-reviewed literature. This process took 15 months from start to finish. So the key steps to our data analysis include: the working group discussed the approach and developed an analysis plan, and then the drug sponsors uploaded the patient-level data from their clinical trials. CRAB performed the analysis based on the analysis plan, And then the working group reviewed and formulated findings. Our meta-analysis included 5 clinical trials. This was 200 patients with non-small cell lung cancer who were treated with immunotherapy. And so what we did was categorize those patients based on a change in ctDNA. We had 63 patients with a strong decrease in ctDNA levels from baseline. So baseline in this case is before they received any treatment. We had 103 patients with an intermediate change in ctDNA levels from baseline, and 34 patients had a strong increase in ctDNA levels from baseline. And so we were interested in understanding the overall survival within these 3 groups. And so this Kaplan-Meier plot, which is on the right, displays the probability of survival over time. And what it shows is that patients with a strong increase in ctDNA from baseline have the lowest overall survival. Patients with the strongest decrease in ctDNA from baseline have the highest overall survival, and patients with an intermediate change in ctDNA from baseline have a moderate survival. And so we revisited our key questions and tried to figure out if we were able to answer them during our meta-analysis. The first question, do ctDNA levels reflect therapeutic effect in cancer therapies? The answer is yes. Reductions in ctDNA are strongly associated with better clinical outcomes. And second, can we bring data together? Also yes. Harmonizing data across data sets is possible with collaboration. These findings set the stage for step 2 of our project. This phase expands into more patients, more tumor types, and more treatments. We have 22 clinical trials representing over 3,000 patients with 8 tumor types and 16 therapies. And these patients were broken into 3 different modules that we'll analyze in a meta-analysis individually and then pull across. Module 1 includes non-small cell lung cancer treated with tyrosine kinase inhibitors or TKIs. Module 2 is similar to what we saw in step 1 with non-small cell lung cancer and immunotherapy, but we also have a chemotherapy arm in module 2. And module 3, which includes solid tumors and either immunotherapy or TKIs. And this phase is currently underway. So our key takeaways of what we've talked about here today are that ctDNA holds a lot of promise as a tool to understand cancer. But in order to use it as a tool, we need to establish data and evidence to fully realize the potential. And Friends is helping to make this happen. At this point, I'd like to pass it over to our panelists to discuss and take questions from the audience. So you can use the question and answer function through Zoom to ask questions. We have a few questions that will get started, but feel free to send a question at any time. So I'd like to introduce Dr. Mark Stewart, from VP of Science Policy for Friends of Cancer Research, Chris Abish from AstraZeneca, Kristen McJunkins, who is a patient advocate, and Paz Valanki, who is from FDA.
Thanks, Hillary. Thanks so much for making time to participate in the discussion today. And thanks, Hillary and Brittany, for that helpful overview of ctDNA and some of the near-term applications and how, as an organization, we're helping generate some of the necessary evidence to really fully realize the use of ctDNA. As Hillary mentioned, we've devoted much of the meeting today to hear from you all and certainly encourage you all to submit questions. And while those are coming in, thought I might just start with some general questions for our panelists to get things started. So I'm curious, the presentations acknowledged a number of applications of ctDNA. From each of your perspectives, I'd love to hear kind of what kind of excites you most about ctDNA, and particularly the impact it can have on patients with cancer. And so I might just start with Chris for that.
Yeah, sure. I think that's a great question, Mark. There's obviously a lot of movement in the ctDNA space, and there are lots of different applications of these technologies being considered, some of which we've just covered. I think particularly from my perspective, one very exciting application is using circulating tumor DNA to detect cancers early. So for example, can you look for these very rare fragments of tumor DNA in the blood of people who don't have symptoms from cancer, and uncover undiagnosed cancers at an earlier stage. So I'm watching that space with a lot of interest to see what happens there. But I do think some of the topics we've just covered in terms of using circulating tumor DNA to monitor response to therapy is very exciting from a drug development perspective, because we're obviously developing a lot of new drugs for cancer patients, and we see some patients have excellent responses to those drugs, but we need mechanisms to have surrogates for how those responses match to those intermediate and late endpoints like overall survival. And circulating tumor DNA is really attractive from that perspective, because it's an analyte that we can quite easily monitor in patients. It's an easy blood test. And if we can generate an evidence base that tells us, yes, if we have a patient in a trial and they've got a clearance or good reduction in their levels of circulating tumor DNA, that those patients are going to go on and do very well when we follow them up for survival at 1-2 years' time, then that gives us the capability to provide evidence to regulators that yeah, let's try and make these drugs available faster for patients so we can increase access to them, rather than patients having to wait for 2-3 years, despite us seeing these really good responses very early on. So from my perspective, I think that's also a super exciting application.
Wow, Kristen, what are you hearing from your fellow advocates, and what excites you about the potential of ctDNA?
Sure, thank you for having me. And just to give a little background, I'm a melanoma survivor, 14 years. And when I was diagnosed, I didn't actually qualify for any clinical trials. But I've stayed involved with my support group over the years. And so, I've seen a lot of people be part of the drug development that's happened. And also, full disclosure, I just really learned about ctDNA 3-4 weeks ago. So I've been trying to do as much reading as I can about it. And from a patient perspective, I think it's really exciting, as Chris mentioned, that there can really be, can often precede clinical signs of disease, and that it really is has great potential as a screening tool early on before things progress too much. And then I think also, seeing so many people that have had different levels of toxicity with clinical trials and things like that, to really be able to determine earlier on whether it's actually working or making any progress, or if a different course of action needs to be taken more quickly, to help relieve some of those side effects.
And Paz, you certainly have your finger on the pulse and can probably speak to some of the trends that you're seeing in terms of how it's being used within drug development. I'm curious kind of what you're seeing.
Yeah, just to add on to what Chris and Kristen said, we're really excited about all of the clinical uses of ctDNA. But as you've mentioned, there's a lot of regulatory uses of ctDNA that we're using in clinical trials, and that we're seeing and hope to see help make drug development more efficient. I believe, Brittany and Hillary already talked about our FDA guidance and some of the potential regulatory uses of ctDNA. But to again kind of highlight, we're using ctDNA to enrich patient populations in clinical trials so that we can potentially maybe don't have to have as large of clinical trials, and we can get trials done or completed earlier to potentially expedite drug development. That's our big hope. It's also being used to select patients for clinical trials, and as Chris mentioned, to identify patients that are likely or hopefully likely to respond to therapies. So there's a lot of interesting and important ways that it's being used to expedite drug development. And I think we'll probably touch on this more. But again, I know that there's a lot of interest too about using ctDNA as an earlier endpoint in clinical trials, again, also to bring therapies to patients quicker. And so there's a lot of excitement, but more work needs to be done there in terms of generating evidence to support that as an early endpoint, but a lot of interest in the regulatory community as well.
Thanks. And we're getting a number of questions in, and Chris, you touched on this in your comment around the potential for using ctDNA to even identify patients that have cancer. And in fact, a question came in regarding that potential use. I'm wondering if you might be able to speak a bit more in terms of where we are in reaching that goal, and what's left to do.
Sure. So we're certainly not quite at the point where we're certain that using circulating tumor DNA can help us, diagnose patients earlier and diagnose cancers at an earlier stage. But I think we're well along that path. So one assay that we're quite excited about following is an assay that's made by a company called Grail. And they've developed a blood test that can look for 50 different types of cancers in your blood. And they do that by looking for fragments of DNA that have something called an abnormal methylation signal. So the methylation signal on a piece of DNA tells you where that DNA is coming from. So their test can detect this abnormally methylated DNA that's indicative of cancer. And it also tells you, well, this might be a lung cancer or it might be a bladder cancer. So they've done a lot of work developing this test, but they've only characterized it in patients who've been diagnosed with cancer mainly. They've done small studies in screening contexts. But in the UK, they've embarked on a very ambitious study in our National Health Service that's actually designed to say, well, if we detect cancer in screening populations using this test, are we detecting cancers at an earlier stage? And I think it's that kind of evidence that will move us along the roadmap towards implementation of these technologies. And that's the first step, can you detect cancers earlier? And then really you want some indication as well that detecting these cancers earlier result in benefit to the patients. So it's about generating that data to give us the confidence to use the tests in that fashion.
So another question came in specifically related to our CT Monitor project and some of the mechanisms for conducting our meta-analysis. And I might rephrase it just slightly, but the question was really getting at, what types of ctDNA specifically are we measuring? Were all the trials using the same ctDNA collection methods and tests? I might rephrase that because I think it highlights kind of a challenge that as a field we're having to grapple with right now in terms of the different types of technology that are being used, the different types of ways it's being integrated in clinical trials. And I think that certainly does create challenges when we're trying to make sense of these different data sets that are coming from clinical trials. And so I might ask Chris and Paz potentially to maybe discuss some of those challenges and why it's important to work through some of those now as a community in terms of creating some standards and harmonizations for how this technology is being used.
Do you want to go first, Paz?
Sure. Yes, I can go ahead. So that's a great question. So we have to think about ctDNA across different tumor types, for example, and we want to think about how we can analyze existing data together, for example. So there's different biology of ctDNA potentially depending on the tumor type, depending on stage of disease, depending on what type of therapy a patient may receive. So we need to think about all of these different disease characteristics and parameters when looking at the data. As you mentioned, there's also differences in types of assays that are available. Are the assays, is it a tumor naive assay, meaning is this an assay that we don't need to have prior information from the patient's tumor to use, or is it something that we're using information from the patient's tumor to make decisions or to make an assessment. So we need to think about all of these different parameters and how trials have been looking at these things when we're pulling together data. And I'm sorry, I think, I'll pass it over to Chris. I'm not sure if I've lost track of the question, but essentially, there's lots of different things that we need to look at in terms of differences across tumor types.
Yeah, no, I think that covered partly what I was going to say as well. Is essentially, I think we might view it, we might have initially thought it would be simple that we could take these data from multiple trials and try and analyze it together to create evidence to guide our use of ctDNA. But I think as we understand more and more about the science behind circulating tumor DNA, it's obvious that there are some tumors that release little amounts of circulating tumor DNA, where taking this approach might be less useful. As you mentioned, the stage of the tumor as well. So we heard it mentioned that the amount of circulating tumor DNA in your blood varies with the size of the tumor. So it may be more challenging to use circulating tumor DNA in contexts of very low disease burden. And then from the technology side, there's a huge array of different technologies available to measure circulating tumor DNA. So we also need to consider some form of standardization. How do we say that one test is comparable to a second test? So we're trying to take all of these factors into account when we look across trials to draw conclusions about utility of circulating tumor DNA.
I feel like I'm loading you up with questions, but another question that came in was kind of gets to the point of the applicability of ctDNA and multiple cancer types. And I think a lot of our discussion today and even our project's been focused on solid tumors, but the truth is, there are applications of ctDNA and blood cancers as well. And in fact, I think the use of ctDNA was, probably initiated there or is a little bit further along. Could you just maybe just briefly kind of mention some of those applications and even, how we can even position some of the learnings, in that setting to what we're trying to do in solid tumors?
So I can go ahead and start. So you're absolutely right that a lot of the use of ctDNA was actually used earlier with hematologic malignancies, and there's been more experience in that space. There's actually been FDA guidances on this as well. And there's been a number of workshops over the years on this particular topic. And so there's more data in the hematologic malignancies field compared to solid tumors. And so we're looking to the experience with hematologic malignancies a lot as we're trying to move this forward with solid tumors as well. So absolutely, it is being used with hematologic malignancies. In fact, there have been even some situations where we've used, where we've monitored minimal residual disease for hematologic malignancies, and we're using that as a way to kind of help support an approval of drugs. And that's in the early stages as well, but that is something that has been done, and that's not something we've done yet with solid tumors.
Let's see. So one other question that came in is around the use of ctDNA for maybe patients that are in remission. So we've talked about how ctDNA could be used to identify patients with cancer, how it could help enrich patient population or enrich for patients that are likely to progress. So I think expanding on that a bit in terms of how ctDNA could be used for patients that are kind of in this remission period.
Yes, so I'm happy to take that one, Mark. So this is another quite interesting application of the technology. So what we've seen, for example, in lung cancer, is that you can use circulating tumor DNA after a treatment that might be curative to identify patients who have residual disease after that treatment. So what that means in practice, for example, is that someone has a lung cancer that's diagnosed that can be surgically resected, that tumor is removed, and then they give a blood test, and we look in that blood test for ultra low levels of circulating tumor DNA that indicates that unfortunately somewhere in that person's body is a site of cancer that wasn't removed by surgery. And the application of this is actually quite groundbreaking, because what we currently do as oncologists in the lung cancer space is that we say, well, we've removed this patient's cancer, we've looked at it under the microscope, we do something called staging, and we say those patients are at high risk of disease recurrence. So if a patient is stage 3, for example, we might suggest that in the region of 50% of those patients might go on and suffer disease recurrence. But what we can potentially do with circulating tumor DNA is improve on that dramatically and say, well, if we detect ctDNA in the blood, the positive predictive value, so the value of that test for predicting recurrence, is above 90%. So it means that there's definitely some disease there. And that creates opportunities to try and cure those patients with more aggressive drug treatment. So can we escalate their treatment after surgery to try and eradicate that very low burden disease? And we do see cures in this post-surgical setting. Our historical trial data in lung cancer shows us that if we give chemotherapy after surgery, we can cure patients. So I think that creates a very exciting opportunity to innovate treatments in. And then there's also opportunities to say, well, if we don't detect circulating tumor DNA, do these patients require additional treatment? So de-escalate treatment. And I think that's more challenging, because you need to be really certain that the circulating tumor DNA is not there. But we saw some very exciting data earlier this year in colorectal cancer suggesting that might be feasible in the near-term future. So this use of circulating tumor DNA, I think, is something to watch as well.
And Kristen, I have a question for you. There's certainly implications around the burden on patients when it comes to not only participating in clinical trials, but just burden associated with having to go into the clinic and receiving various cancer therapies, particularly if it's an IV treatment, for instance, and having to go to outpatient care. How might you envision ctDNA being implemented, and does it kind of present an opportunity to kind of facilitate or make it somewhat easier? If you're looking at a simple blood draw, for instance, compared to clinical imaging.
Yeah.
Sure. What if you could speak to even some of the experiences you had and how ctDNA might alleviate or help with some of that?
So it's another interesting discussion point. So currently, standard of care after surgery is that you would come back to your hospital every 3 to 6 months and have a scan by one of these CT machines that would look at your lungs and other sites of the body to see if the lung cancer is coming back or any cancer is coming back. Now, we've seen in lung cancer, and it's been shown in breast cancer, colorectal cancer, bladder cancer, that actually a blood draw can also detect cancer coming back as well. So there's potential there, right? Because you could, instead of advising a patient to come in every 3 months in the first year for a CT scan, some of those visits could alternate with donating blood at your pharmacy for a blood test to see if there's any level of detection of cancer. And I think there's also economic consequences there as well, because you're potentially not using as much resource in terms of scanning, having a radiologist interpret the scans, etc. So that's another interesting argument for this. I think it's important to point out that I don't think these blood tests would replace imaging, because then they're not going to tell you definitely if the cancer's back. I think you'd always, as a physician, want to confirm that on imaging, and the imaging as well gives you the anatomy and tells you where the tumor is if it's recurring in the body, so that doctors can plan further treatment. But I definitely think there's potential for these blood tests to complement that surveillance after surgery. Last point, they're not perfect, the blood tests, because we certainly see that there are some sites of disease relapse that they don't pick up very well. So for example, if a patient's unfortunate enough to suffer a disease relapse in the brain, the blood tests sometimes don't pick those up. We think that might be because of the protective layer of the brain called the blood-brain barrier, which prevents DNA going into the blood. So I think that just emphasizes that there's still a lot of work to do to provide guidance. You know, how should we be using these tests? Where can they be useful? What scenarios might you want to use imaging, for example, like intracranial or head imaging? So a lot of potential, but more work to do to work out the best way to use this in the clinic.
And I just wanted to add, as Chris was talking, I completely agree, but I was also thinking in terms of, particularly if people have been determined no evidence of disease or in remission, when we think about access to care, I mean, we know that just from a clinical trial standpoint, it's very limiting as to who can participate depending on where they live and economic status and things like that. But, if they're in remission and live in a more rural area, and that, instead of every 3 months having to go for imaging, maybe every 6 months, and doing the blood test in between, that might increase continuity of care for people that don't have access on a regular basis.
Yep. No, I think that's a great point. We certainly have patients on our trials who have to have air travel to go to their site to be followed up. So any way to kind of alleviate that pressure on patients is obviously going to be helpful.
It's really helpful to hear. I certainly could remove some of the instances of watchful waiting, I can imagine, and helping provide a more objective or more clear insights in terms of what to do in those periods of remission. So it was kind of briefly touched on around kind of what if a patient does have no evidence of disease after receiving treatment, and the cancer is not visible in current screening methods, but say you do detect ctDNA, this is probably still requires additional kind of evidence and data to really understand kind of the best approach here. But would treatment be recommended before the actual disease presents itself as a recurrence?
So I can start. So I think we need to be careful about recommending additional treatment at this point outside of a clinical trial, just based on ctDNA results alone. We don't know exactly what that means all of the time. You also, on the other hand, need to think about, is the ctDNA result providing additional information compared to imaging scans? It's possible that ctDNA result might shortly translate into development of disease on a scan. So that's something to think about as well. But there are clinical trials that are looking at making treatment decisions based on the presence of ctDNA, particularly in the adjuvant setting. And I think that's going to be a really important area of research to further explore the use of ctDNA in this capacity.
Yeah, if I could add to that, I think no test is perfect, right? There's always going to be some tests that come back positive as when there's not cancer, even though it's quite rare with these type of ctDNA tests. So I think that's always a consideration that physicians have when they see a positive result without disease on obvious or disease on the scan. So as Paz said, ideally you'd want evidence from trials that show you if you have that specific clinical scenario and intervene, you're helping patients in terms of preventing disease relapse or prolonging disease-free periods. One other aspect that's relevant to this is there's a lot of work to show that actually ctDNA can help when physicians or radiologists aren't sure of what they're seeing on a scan. So radiology itself isn't black and white. You certainly see things after surgery on a scan, for example, that you want to follow up over time to see how they change, because they could be an infection or just the anatomy of the chest after surgery. And circulating tumor DNA could be quite helpful there, because the radiologist could be concerned by an appearance, but say, hang on, I want an extra test to give me confidence that this is tumor. And that could lead to help physicians to intervene earlier, because they could be more certain earlier if they see a lesion on a scan that could be cancer, but the patient's blood is positive, then that may lead you to intervene earlier than you would if you just had imaging alone, and that could help patients by bringing forward curative interventions like radiotherapy, for example.
Well, I think we've made it through most of the questions that have come through, and certainly don't want to kind of belabor any points here, but this has been a really insightful and helpful conversation, and certainly appreciate all that each of you are doing to help advance this. I think it's certainly important to acknowledge the reason why we're all kind of striving to do this in order to improve lives of patients with cancer, and making sure that as we're generating the evidence and integrating this into trials, that we keep that perspective in mind and are working towards that common goal. And again, appreciate all the different efforts that are going into that from each of you all. I guess maybe before I wrap up, I don't know if there are any kind of final comments that any of you want to make related to ctDNA that you didn't get a chance to, just to wrap this up. If there are any kind of big takeaways and
I mean, I'll just start from a patient perspective. I think having more tools in the toolbox for physicians, I mean, obviously with patients, the anxiety and the stress of everything, and certainly the watch and wait. And if I know from my perspective, I'm thinking about post-treatment, I was no evidence of disease, but I think it was Chris that had mentioned, if there was, instead of having scans every 6 months for 5 years like I did, if I could do blood tests in between, just to kind of help alleviate the fear and the anxiety that comes with that. Or even when something is found and it is a watch and wait situation before deciding on any treatment, if you can intermittently have some of this, because again, maybe if you can find out that it's progressing quicker than what a scan is going to show every year, that can help alleviate some of this stress and get patients into treatment quicker.
Absolutely.
I guess to add on to that, I'm very excited about this space. It's really wonderful that this is a less invasive tool compared to doing a tissue biopsy, for example. And so this is something that can be done longitudinally over time. We can look at the genetics of tumors over time to see how patients are responding to tumors, and if there are specific drug resistance mechanisms that are developing, and help us, plan our next attacks. So I think there's so many important uses for ctDNA, and I think it's really heartening to see all of these efforts across academia, industry, Friends of Cancer Research to try to really generate quality evidence to really support the use of ctDNA further to really support drug development.
Yeah, and if I could just add, I think one theme when we've been answering these questions, whether it's related to screening or detecting residual cancer after surgery and intervening, etc., is that physicians need evidence to make decisions with, and that we need evidence also to support claims based around circulating tumor DNA as a marker that a patient's responding to treatment. So these endeavors, like the one we've discussed earlier, the CT Monitor project, where you're bringing together data sets from multiple different partners, industry, from academia, are really valuable, because I think all of the patients who've gone into these studies and donated samples have done it because they want to advance research as well as be involved in the trial, and it's so much more powerful when you pull together all of these different data sets to create these evidence foundations to guide practice, and so it's just to say it's very valuable what Friends of Cancer are doing.
Thank you, and thank you all. We're excited to be on this journey with you all. We certainly recognize the potential of ctDNA and the impact it can have on oncology drug development, and we're just excited to be at this point in our CT Monitor project, and know that there will be multiple kind of data readouts and meetings that we'll be having into 2023 as that data comes together, and as a community we work to better understand what it's telling us and how best to implement it moving forward. So with that, thank you. Thank you for those that attended and participated. Really appreciated all the great questions that came in, and hope we have additional opportunities to do this again soon.